Okay, students, so now we are going to start a mock interview for our student Aishwarya, and this mock is for biochemical engineering in IIT BHU. So Aishwarya, first of all, you introduce yourself.
Good evening, sir. Good evening, ma'am. I'm very grateful for this opportunity. I'm Aishwarya Jaishankar from Kallur in Tamil Nadu. I have done my undergraduate degree, B.Tech in Industrial Biotechnology from Government College of Technology, Coimbatore with 8.59 CGPA. I'm very much passionate about biotechnology, and I believe that the very passion has taken to me this place. Not to lag behind, I enrolled myself in a few clubs in my college to develop my interpersonal skills that helped me a lot during my final year project to be a better team player. I did my final year project on the topic of lignin nanoparticle synthesis from the groundnut shell and checked for their antimicrobial activity. I have also participated in inter-college competitions like Smart India Hackathon, and I also did one theoretical project on the topic of what wastewater treatment at the household level by the use of hibiscus cannabinus. I have always wanted to fulfill my dream, and that has driven me to this place. Thank you, sir.
Okay. So Aishwarya, tell me, why do you want to join IIT BHU?
So because from the early stages when I joined biotechnology, I have my own interest on the bioprocess system and design. So I think biochemical in IIT BHU, they have very good work on the bioprocessing. So I want to join in IIT BHU Biochemical Engineering. Sir.
Okay. So did you did you find any suitable research area here or any particular professor?
Yes, sir. I saw one professor profile with bioprocess design, and he do some scaling up process in bioprocessing, sir.
Okay. So you are ready for PhD also?
Yes, sir. In future, maybe.
Okay. This time, if we offer you PhD, then?
Yes, sir. If time constraints, I will definitely accept this, sir.
Okay. So what is your long-term career goal?
I want to become a professor, sir. That is my career goal.
Okay. Can you tell us any subject in mathematics have you studied?
Yes, sir. I studied linear algebra and probability during my GATE and also during my undergraduation, sir.
Okay. So tell me, what is the meaning of independent and dependent events?
Variables, sir?
Independent and dependent events. What is the difference?
So, if A and B are the two events, if the event B will depend totally upon the event A, then it is called the dependent event. If there is no correlation between the A and B, then they are called as the independent events. That means the probability of A intersection with B will be totally zero.
So if A and B are independent, then how will we write P A intersection B? Can you write it in the notebook? Do you have some notebook?
Yes, sir. That is P of A bar B. That is a conditional probability, sir.
A and B are independent events.
Yes, sir.
So what will be the value of P A intersection B equal to? What will you write it?
P of A into P of
A intersection B equal to?
Yes, sir. I think this will suit, sir. This is clear, sir?
P of A intersection B is P of A into P of A upon B. Now I am saying A and B are independent. Then
I use the conditional probability here, sir. In that I studied like P of A given B. If they are independent, even we can use like this.
So finally, what will it reduce to? Do you know random variable?
Yes, sir.
What is random variable?
If we, for example, sir, can I explain with the example, sir?
Yes, yes.
If we have a coin, so we have tossed, we can get head and tail. So we can't able to give the probability for the head and tail. It should be we have some numbers. For that, we created the random variables. Here we can able to say that the probability of head will be 1 and the probability of tail will be 2. For that use of the numbers, we use the random variables.
Okay. Any example of random variable related to your core area?
Related to my-
Take
Second
any
base.
example of random variable related to your area, means your biotechnology field. Anything you can take.
So I think there are many applications maybe, but not sure what that is. But I sure many applications there are.
So not getting any particular example right now.
No, sir.
Okay. And do you know about transducers? Have you studied transducers?
No, sir.
Chemical sensors?
No, sir.
Okay. Okay. Barsa ma'am, you can continue.
Hello, Ashwarya.
Good evening, ma'am.
Good evening. Ashwarya, my first question is to you is, what is genetically modified organism?
Yes, ma'am. If we want to create a genetically modified organism, if we have any gene of interest with us, then we are going to do cloning and expressing it on the another organism that is called the host. So when we do like that, then finally we can able to get the genetically modified organism, ma'am.
Okay, give an example.
Basically, Dolly is a no, ma'am. Plants. We have plants, ma'am. Genetically modified tomato.
Only
and more-
plants. Only plants, not animals and not microorganisms.
Yes, ma'am. We have transgenic cows and sheep and fish
Okay.
also, ma'am.
Good. And an example of microorganism?
Modified microorganism, ma'am.
Yes, GMO.
Yes, ma'am. We have like a modified E. coli also for the benefit of cloning and expression.
In case of agriculture, if I ask, in case of agriculture.
Yes, ma'am. We have more that Bacillus thuringiensis and more and more things in case of agriculture.
Agrobacterium. One is agrobacterium.
Yes, ma'am. Agrobacterium.
Okay. Acha, what is sterilization?
That is, we are going to do sterilization for the purpose of contamination, ma'am. So we want to get rid
For
of the
contamination or for to remove contamination?
Yes, ma'am. To get rid of the contamination, we are
Okay,
going to do sterilization.
Okay. What are the different type of sterilization?
Ma'am, in small scale, we can do autoclave, ma'am. But in the
Okay.
large scale, we can do heating. And with the use of heat exchangers, we can do sterilization. And
Okay.
coils, internal coil, external coils are there for sterilization. And also UV we can use, ma'am.
Okay. Acha, my first thing is, what are the different conditions in the case of autoclaving?
Different conditions, ma'am. The pressure should be 180 mm, ma'am. And the temperature should go upon 121 degrees Celsius, ma'am.
Okay, or in case of heating, if we use any heat object, then like autoclave, not autoclave, like incubator, then what are the different conditions?
In incubator, we will choose the condition based upon the organism we are going to using, ma'am.
Okay.
Depends upon.
If we sterilize glassware in hot air oven, then what are the temperature and timing for sterilization?
Ma'am, I think I used incubator with the glass. So I use a maximum degree Celsius of up to 37 to 40 degrees Celsius, ma'am.
No. This is normally for the growth of microorganism. 37 degrees normally growth of for sterilization purpose. Temperature should be around
100
160 degree. 160 degree.
Okay, ma'am.
Okay, for 2 hours.
Okay, ma'am.
Okay. Next question. In the case of laminar flow, which kind of UV light is used?
Blue UV light, ma'am.
No. Which wavelength? Which type of UV light?
Okay.
UVA,
That is
either
in the-
UVA, B or C.
I think UVA, ma'am. That is in the range of 73.
UVC.
Okay.
UVC. Okay, recall all these things.
Okay.
My next question is to you. What is Beer-Lambert law?
Beer-Lambert law, which basically used in the UV spectrometry, ma'am. That is A, which is equal to epsilon into C into D. A basically the observance, epsilon is the coefficient of that, and C will be the concentration of the solution. D will be the diameter of the cuvette which we used.
Okay. You hear about sigma subunit in the case of prokaryotic transcription. What is the role of sigma in prokaryotic transcription?
Yes, ma'am, there is one sigma factor which will allow the RNA polymerase to sit on that and continue the replication. Without the sigma factor, we can't able to start the transcription process, ma'am.
Without sigma, okay, good. Lactose is monosaccharide or disaccharide? Lactose, disaccharide. What are the different components in lactose?
Glucose and mannose.
Glucose and?
Mannose. Sorry, ma'am, glucose and galactose.
Galactose. Or in case of sucrose?
Glucose and glucose, ma'am.
Glucose and glucose. Glucose, chicken. What is formula for molarity?
I only remember the denominator, something volume of. Sorry, ma'am, I can't remember that.
Okay. Molarity is weight of solute upon molecular weight of solute, and finally volume of solvent in liters. आप इसको calculate करोगे, then you get molarity. In case of water, what is the value of molarity in case of water?
I remember approximately that is 1.16 or
55.5. Okay. You heard about ethidium bromide? What is the role of ethidium bromide in
DNA staining, ma'am?
Okay.
Electrophoresis method, we basically use ETBR as a staining solution.
What is ethidium bromide?
Ethidium bromide basically we use it is going to be a chelating agent. In case of electrophoresis, we are going to use as a chelating agent, and for the staining solution, it will bind to the DNA bases, inter between the DNA bases, and it is going to look like a orange color under the UV.
Okay, good. You hear about bromophenol blue and cyanol.
Yes, ma'am. That is also used in case of electrophoresis, and they are basically the leading dye and the lagging dye. Basically, bromophenol blue is used as a leading dye because of the smaller size of the bromophenol blue, and xylene, which is going to be used as a lagging dye. So these are all the dyes which will be used to track the electrophoresis, ma'am.
Okay, good. Which kind of biomolecules are separated in Western blotting or identified in Western blotting? Which kind of biomolecules? Proteins. Okay. In case of Southern Western blotting?
In case of Southern Western, we use DNA and protein, ma'am. DNA and protein interaction.
DNA binding protein. An example?
Technique, ma'am. Any DNA protein interaction? We have transcription factors with the DNA, ma'am.
Yes. Okay. Good. What is Taq polymerase?
Taq polymerase is a very important enzyme in case of PCR technique and many other DNA sequencing techniques. Because of that high temperature, it can able to tolerate. So it have the capability to. It don't have any capability for proofreading, but it can able to 5' to 3' exonuclease activity, ma'am.
Okay. Okay. What is the site of trypsin endopeptidase? Trypsin endopeptidase. What are the different amino acid which are acted upon by endopeptidase?
I don't know.
Okay. Lysine and arginine. Okay. What are different type of hypersensitivity?
4 different types of hypersensitivity are there, ma'am.
Name them.
Type 1, basically type 1 and type 2 antibodies will play the role, and then type 3 the complement will play the role. In type 4 T cells will play the role.
That's good. Which kind of antibody play a role in case of type 1 hypersensitivity?
Antibody E, ma'am. Antibody E and mast cells will play the major role in type 1 hypersensitivity.
And in case of type 2, which kind of antibody?
Type 2, the activation of the ADCC complex, antibody dependent complex will activate, ma'am. Complex and the antibody. Complement
IgG antibody.
IgG.
Okay, okay. What are different excitatory neurotransmitters? Excitatory neurotransmitter. Give an example of excitatory neurotransmitter.
Don't know.
Okay. Name the pacemaker of glycolysis.
Huh?
Pacemaker of glycolysis. Any enzyme which is responsible or which act as a pacemaker of glycolysis.
Hexokinase, ma'am.
Which?
Hexokinase.
Okay. Give an example of G protein coupled receptor.
The G protein coupled receptor will be used in cases of sensories and rhodopsin in eye and sensory elements and taste buds also. In all these cases, G protein coupled receptor
are used. Okay. My last question is to you is, what is in vitro and in vivo techniques?
In vitro means first in vivo, it is a technique. We are going to do the live natural process outside in the laboratory. In vitro means we are going to do all these techniques within the living organism.
You are sure?
In vitro outside the laboratory, in vivo inside the organism.
In vivo inside the organism.
In vitro outside the laboratory.
Okay, okay. In vitro means inside the tube.
Okay.
Same condition as in case of organism. Same condition follow in case of in vitro studies. Okay. Okay. Thank you, Aishwarya.
Thank you.
Okay, Nikhil sir.
Okay. So Aishwarya, how are you?
Thanks, sir. How are you, sir?
I'm also good. Okay. Thank you. So my first question to you: what is the difference between a batch and semi-batch reactor?
Basically, batch, we are going to give one input and final going to get one output, sir. But in case of semi-batch, we can either going to give one input or we can able to get output, sir. That depends upon our application. We are going to choose, we want to going to give continuously the input or we are going to take continuously the output, sir.
Okay. So where do we generally use the batch reactor? What are the major applications of batch reactor?
The major application of batch reactor we can use in case of enzyme culture, sir. It is best to use in case of enzymes. When we use immobilized enzyme or when we use enzymes, batch reactor going to be the serve the best, sir.
Anything else?
And also in cell culture, if we want to take any secondary metabolite, we can use batch culture, sir.
Okay. Okay. Tell me, what is fermentation?
Fermentation means we are going to some organisms do the fermentation. Basically the chemical reactions which is going to take place during the fermentation reaction. The final product we can get fermented.
Define me what is fermentation first. In a simple word.
Okay, sir. It is a process done by some microorganisms in any element. That's it, sir.
Okay, no problem. Okay, tell me, what is the difference between chemical reaction and enzymatic reactions?
So chemical reaction can be done by any catalyst or any chemicals, but in case of enzymatic reactions, enzyme will play the role and enzyme have their own active site. They are going to in the physical contact, but in case of chemical reactions, that always not matters.
How?
So
How they are not in physical contact in chemical reactions, catalyst and reactants?
So they have one, in case of enzymes, they have one particular site called active site. In that particular site, they should be in the physical contact. Otherwise, they are not going to be in work. But in case of chemical reactions, we can just mix it and we can get the results.
Okay. Anything else?
No, sir.
Okay, no problem. Okay, tell me how to prevent vortex formation in a batch reactor.
Yes, sir. For that, we have structures called baffles. So baffles are very important to prevent the over vortex of the batch reactor, sir. And also we have impeller stirrer shaft. So these are the components basically going to reduce the vortex, sir.
Okay. Tell me the second reason once again. What about the impellers?
So the impeller hanging over to the stirrer shaft, sir. So the baffles and the stirrer shaft going to reduce the vortex, sir.
There will be impeller, right, in a batch reactor. Already
Yes,
there
sir.
will be.
Yes, sir.
So impeller will reduce the vortex formation, you are saying.
No, sir. Impeller hanging to the stirrer shaft, sir. That stirrer shaft and the baffles going to do the work, not the
Okay,
impeller.
okay. Okay, what are the different steps in a chain reaction? You know,
Sir,
what are the different steps in a chain reaction?
chain reaction, sir.
Yes.
I don't know, sir. I couldn't get.
Okay, have you heard about Michaelis-Menten equation?
Yes, sir.
Oh, tell me that equation.
V is equal to Vmax into S divided by Km plus S. Basically, V is the rate of the velocity of the enzyme. Vmax is a maximum velocity of that enzyme. S is a substrate divided by Km will be the Michaelis-Menten constant.
Okay. What is the unit of Michaelis constant?
Michaelis-Menten constant, sir. What is the
Yes.
unit for a substrate we are going to give? The same unit will be for the Km, sir.
Okay. And what is turnover number?
Give me seconds, sir.
Okay, take your time.
It depends to the catalytic activity and also the. I can't able to give it fully, sir. I know, but
Okay, no problem. Okay, sir, I am done from my side.
Okay, sir, provide feedback.
Okay, so Surya, as far as your fundamental knowledge goes, your fundamental knowledge is good enough. And just go through the some major topic like design of scale of a biological reactor, bioseparation processes, treatment of bioprocesses in like plant design. Okay, and the reactions like Michaelis-Menten reactions. Okay, different denominations like terminology we are using here, and with including its unit. Okay,
okay.
and rest is fine, I think.
Thank you, sir.
And one more thing I would like to add, what is the difference between chemical reaction and enzymatic reaction?
Yes, sir.
So you can say that like enzyme catalysts are highly specific. Okay. Apart from this, the rate of enzyme catalyzed reactions is usually much faster than the chemical reaction.
Okay, sir.
Okay. And the reaction conditions are mild. In enzymatic reactions, we generally use normal temperature pressure, isn't it? And in chemical reactions, the temperature will be much higher. Okay. And enzymes are comparatively very much sensitive, isn't it? So you can just differentiate like this. Okay.
Sure, sir.
And what is fermentation? You can say that it is an enzymatically controlled transformation of an organic compound. Okay. You can say like that. Okay. Over to you, Tyagi sir.
Thank you, sir.
Okay, Varsa ma'am.
Aishwarya, yes, your delivery is good, and your subject knowledge also fine to good, but you should work on some area like instrumentation and their importance. technique like electrophoresis, sequencing, mass spectroscopy, chromatography, and ELISA. Okay, and different kind of receptor which are used for is question for you. Receptor used for
what? I can't get it clear.
different kind of receptor used for
Signaling, ma'am.
where we can use receptor.
Receptors we can use. Receptor is a very important in our human body itself, ma'am.
Okay.
For signaling pathways and all.
Yes, in all the signaling pathway which are usually happen in case of cell, so receptor play a very important role. Okay, you work on receptor. You have very little knowledge about receptor, and I think many questions may be asked from this area. Okay, work on this area, and all the best for your exam.
Thank you.
Okay, for your interview.
Okay, Swarya. Probability you did not answer correctly.
Okay, sir.
So work on probability random variable. Okay, they can ask questions, and then also study about transducer.
Okay, sir. Sure.
Just go through about sensor, what is sensor, and what are different sensor used in biotechnology, biochemical science, chemical sensor especially. So just go through these topics, then it will be beneficial for you. Okay. Rest your presentation is okay, way of answering is okay, everything is fine. Just work on some more content.
Sure, sir.
Okay, all the best.
Thank you, sir.